Good afternoon. Yeah, as you heard, I'm Damaris Matoke. I work for the Kenya Medical Research Institute as a research scientist. My presentation will be slightly different because at the moment we don't have a genetic drive or genetic approaches that we're using, but I'll just give an overview of what we have done so far and showing that we're ready for the technology. So for KEMRI, our vision is mostly center to be a leading center of excellence in research for human health. And we are anchored under the vision or the mission of improving human health and quality of life through research, capacity building, innovation, and service delivery. This does not mean that we only focus on products that we assess the human, but even we look at products that human consume that affect their health. So the agriculture, environmental factors are very key in informing KEMRI's decision. So these are the mandates, but I've highlighted 3 of them. One of the first one being to conduct health and biomedical and public health research for human health. And the other one is to build human health research capacity. And the 4th one, which is highlighted there, is to be able to advise Ministry of Health and other partners on the decisions to make around human health interventions and healthcare. So we have around 7 programs. The first program being Biotechnology, which is the reason that I'm here, and I lead this program as a deputy director. Under this, we have vaccine development, we have diagnostic, we have genetic engineering, which is very key now in informing genetic biocontrol and other activities related to modifications of the gene. Then there is also data analysis, which is the tool around bioinformatics. However, there are other programs, which is drug development therapeutics, that's using traditional medicine. We have infectious diseases program. We also have non-communicable diseases research. We have One Health approach, which now also is very key when we look at zoonotics, diseases that are transmitted from animals to human, and also diseases that go through animals, the vector, and also touch the human. The other part is sexual reproductive health, but finally, public health and health system strengthening. Those are the key programs we have at KEMRI. So we usually use this platform in identifying what is key for us. The first thing is priority setting, and this advises the research that we are doing, but also innovations, health solutions. And this is the program, the component that hosts the genetic approaches, interventions, or information that is generated. Then finally, we have production of this technology, which is now forms the pipeline for genetic approaches, and also finally coming up with that products for utilization in the field. So I'll narrow my talk to vector-borne diseases because this is where I've been working for a while now. One of them being a vision, which is supposed to be a world free of human suffering from vector-borne diseases. Vector-borne diseases are a major threat, and this consists of mosquitoes, it consists of sandflies, it consists of schistosomiasis. When you heard of schistosomiasis, is one of them that is caused by a snail. Snails, we have STH, which are soil transmitted helminths. We have many others, which is including elephantiasis, that's lymphatic filariasis, which is transmitted by also a mosquito, and many, many others. We know that globally, including Kenya, 80% of the population is at risk of vector-borne diseases. Therefore, but 17% of the population carries this burden, is affected, or we have mortalities from vector-borne diseases. Malaria is the highest because it's around 90%, causing morbidity and mortality from vector-borne diseases. So I'll skip this, but for information purposes, we have zoonotic infections, and we have what we call anthroponotics. Zoonotics are diseases that come from the vector, that's a mosquito, and then they go to the animal and finally get to the human. So there's a cycle between animal and the vector, which is sandfly, then finally they get to the humans. But when we come to anthroponotics, are those diseases that are transmitted from the vector, then they go to the human. That means from the mosquito, I'm using a mosquito as an example, but we have sandflies and other insect-causing diseases. They move from the insect, they go to the human, go again to the insect, to the human, so that's the life cycle. Those are the ones we call anthroponotic infections. So malaria, dengue, have you heard of dengue, chikungunya? We have yellow fever, those form under anthroponotics. But diseases which we don't have most, but arboviruses, most of them form the zoonotic component, and that's where we need the One Health perspective. Yeah, for malaria, to narrow down, in Kenya it's widely spread, and currently I think we are getting endemicity courtesy of the occurrence of an invasive species, Anopheles stephensi. We have a good population, the yellow part being affected from malaria, and populations being at risk of the disease. Currently we know we have diverse mosquitoes transmitting this disease, starting from Anopheles gambiae, which is the commonest mosquito, but trends are changing. Data is showing that this species is declining as we are getting Anopheles arabiensis, which I've listed the third one, taking over. So we have Anopheles gambiae funestus, we have Anopheles stephensi, and we have Anopheles clusi, which have been reported for Kenya specific that are they are carrying the highest burden of malaria in the country. And if you look at the map, you find that these species are co-occurring. It might not be clear, but they are co-occurring mostly in areas where the disease is endemic. Anopheles stephensi, which is the fourth one I've listed there, is the one that was reported recently that used to be an Asian vector, but now it's moving, has moved to the horns of Africa and also moved down to Kenya in Marsabit and other areas that are arid and semi-arid. So, courtesy of this, and as you've seen that vectors are co-occurring, they're transmitting the disease, we believe that gene drives offer solutions. They're able to provide, to be complementary, and that's why we are not saying that gene drives should be standalone. So we know that there are other tools that are actually being used, so they are able to be complementary. They provide a sustainable solution, and also they have ability to be scaled to even areas that are not being reached. So it's important that we consider this gene drive and other genetic approaches when we are determining control of malaria and other vector-borne diseases. So gene drive research is important as it will provide us evidence-based that we'll be using in forming policymakers and other key decision makers in providing guidance and guidelines in the implementation of this. So it's important to support research and utilize results that are data that is generated from the research that we're undertaking. So this is the entire pipeline. I know Professor Ogoi already alluded to it, but as you can see in each and every step, research is needed, be it to inform safety of the products, as we look at efficacy, as we look at the containment measures, monitoring, engagement, and regulatory. That's the framework that is needed. So it's important that data is generated from the research perspective to inform all these steps, from laboratory stages, which is the settings we have like at KEMRI lab stages, to field setups, from small scale to large scale field releases. So how do our infrastructure look like? At KEMRI, we have laboratories, both BSL2, that's biosafety level 2, and we also have biosafety level 3. These are some of the pictures from the labs. But also of importance is that we have an animal rearing facility. Because this animal rearing facility is that can be used to maintain, if it's mosquitoes that you are rearing, also that can be used for animal challenges, experiments, and also to be, before we move to human-related experiments. So for BSL level 3, this is how it looks like. We have containment, and it has measures even before you access the lab. There are various support, all measures that are put in place that give access control and training. For the insectaries, we have where we rear mosquitoes, but also we have insectaries where we rear sandflies, which are transmitting leishmaniasis, a neglected tropical disease. But also we have facilities where we rear snails, and we do snail challenge experiments. And finally, where we do other mosquito, the arbovirus transmitting diseases. So this is the insectary. This is where we raise the adults, and this is where we raise the young ones of the larval stages. And when we do our experimental efficacy testing, we use the WHO recommended methodologies. I wanted to show this as part of now doing the genetic. We are able to now in country or within the institution to be able to analyze any genetic materials by doing either whole genome sequencing, but also we are able to do a fragment analysis because we have the Illumina platforms, and also we have the Nanopore Oxford. These are facilities, and thank God for COVID anyway, because we are able to set up this because provisions of infrastructure for COVID containment and the screening. So these equipments are very key, and they can support assessment of any modifications that we do on genetic related. I would like to indicate that we also are able to train people both within the institution, but students that come in, but also people that come from other institutions and the Ministry of Health. So we partner with institutions to train both lab personnel, but also field workers. So when we go to counties, we're able to train the county team on even sample collections and identifications. So as far as management and also for containment, we have a sample receiving facility, and this is able to monitor what samples come in, what samples go out, what samples are we analyzing the laboratories, and what data is released and how it's shared. So I wanted to show this as showcasing that in case we are able to start a gene drive or any genetic related, we have fields, this is one of them in Western Kenya, where we are able to do experimental heart trials. And this is in case we need to do any sample modification, we are able to do in the field trials. These have been secured by KEMRI, but we have similar, this is in Kisumu, we have another one in Busia, and we have a similar setup in Kilifi, in Kwale, where we are able to do trials, and should we ever think about a release, we could use similar setups that will help in monitoring. This is data that we generated that informed WHO about use of experimental trials. We also engage communities quite intensely in most of our studies, and not only for gene drive, but this is the ability for us, we foresee this as an opportunity for ownership, but also to create awareness and understanding in the communities why a certain approach has been applied and why it's being used in Kenya or in a certain area in the fight against malaria. So we believe that it's important for us to have a co-planning, co-implementation, and co-evaluation, as this will be extremely important in uptake. So we also have a system in place at Cambridge that looks at the ethics of any study we do, and this also is supposed to support the genetic approaches. It works closely with the Institute Biosafety Committee, which looks specifically on genetic related materials, and they are able to review protocols that are submitted and monitor implementation. This is the process. I won't go into it, but it's very detailed and very clear. Therefore, there's no misuse of subjects or misuse of data or misuse of application of controls. So also we use the National Biosafety National Framework, that's the NACOSTI, to get research permits and research licenses. Thank you.